Welcome to the Deep Dive, where we cut through the noise and get straight to those aha moments in the most fascinating corners of science and technology. You've definitely heard of mRNA vaccines, especially in the last few years, right? But have you ever really stopped to wonder about the incredible science and the engineering that makes them possible? And maybe even more importantly, what else this groundbreaking technology is unlocking? Because it's far more than just a shot in the arm. Today, our mission is to really unpack the intricate world of mRNA therapeutics. We're going to explore how scientists are meticulously designing and delivering these genetic instructions. And not just for vaccines, but for a huge array of medical applications, treatments for allergies, even things like cancer. We're going beyond the headlines today. I want to give you those truly surprising facts about, one of medicine's most rapidly evolving fields.
That's exactly right. And it's fascinating because at its heart, mRNA is fundamentally just, a cell's instruction manual. What's truly amazing here is how researchers have learned to harness it, refine it, deliver these instructions, basically transforming them into potent medicines. And it's been quite a journey, really, filled with brilliant problem solving, overcoming challenges that honestly seemed insurmountable at first. The results, though, are genuinely revolutionary for human health.
Okay, so let's unpack this a bit before we dive into all the innovations. Let's try and get a handle on what mRNA actually is in this therapeutic context. We're talking about making medicine. What are the core building blocks, this biological blueprint? What does it look like?
Right. Well, when we talk about a synthetic mRNA molecule, the kind used for therapeutics, it's actually quite a precisely constructed instruction set. It has several key components. So right at the beginning, you've got the 5-foot cap structure. You can think of this as the start signal for the cell's machinery. It's absolutely crucial for getting protein production started, maintaining the mRNA stability, and really importantly, helping the mRNA kind of hide from our body's immune system. Scientists have found specific cap forms, things like Cap1 and Cap2, are much better because they closely mimic our own body's natural mRNA caps. That signals self to the cell, basically, and helps prevent it getting flagged by the immune system right away. Then, right after the cap, there's the 5-foot untranslated region, or the UTR. Now, this is a segment of RNA before the actual coding sequence starts. It doesn't get translated into protein itself, but it heavily influences how efficiently the mRNA is translated and how stable it is. Next up is the coding sequence, or CDS. This is the real core of the message, the actual instructions telling your cell exactly which protein to make. So for a vaccine, this would be an antigen, like, the SARS-CoV-2 spike protein. For other treatments, it might be a therapeutic protein your body needs more of. After the coding sequence, there's another UTR, the 3-foot untranslated region, again, a non-coding bit that follows the main instructions. This part also plays a vital role in regulating how stable the mRNA is and even where it ends up inside the cell. And then finally, right at the very end, you have the poly-A tail. This is just a long string of adenosine nucleotides, literally a tail, and it's essential. It boosts the mRNA's stability, helps kickstart translation, and acts like a protective shield against enzymes that would normally degrade it. So you see, it's a very precise design.
Wow, okay. It really does sound like an incredibly carefully crafted biological blueprint designed for a very specific job. But you mentioned earlier in its natural or maybe naked form, mRNA had some serious drawbacks for medical use, didn't it? You couldn't just design the sequence and, inject it. What were the biggest hurdles there?
Oh, absolutely. Yeah, early attempts with naked mRNA were tough. It made a lot of scientists pretty skeptical about using it medically. The first big problem was just intrinsic instability. RNA is notoriously fragile. If you inject naked mRNA into the body, it gets chewed up really fast by these enzymes everywhere called ribonucleases or RNases. These enzymes just break down the mRNA super quickly, drastically cutting down its chances of actually producing the protein you want inside the cells, which is just, considered too unstable. And the second challenge, just as big, was innate immune recognition. Our immune system is incredibly good at spotting foreign nucleic acids. It sees them as a threat, like a virus invading. So unmodified mRNA, when you introduce it, it triggers a really strong, often unwanted inflammatory response. The immune system has these sophisticated sensors for foreign RNA, and that reaction would basically shut down the therapeutic effect and cause safety issues. It was a major blocker.
Okay, that sounds like a massive problem. Innate immune recognition. That's a fundamental defense system. It seems almost impossible to get around that. How did science possibly navigate that? Because, normally you want your body to react to a threat, but here you needed it not to react, at least not in that way.
It's actually a brilliant story of scientific creativity, and it really starts with understanding something called nucleic acid immunity. Our innate immune system has evolved these really sophisticated ways to detect foreign nucleic acids, whether they're from viruses or bacteria, or even our own nucleic acids if they end up in the wrong place. Think of them like highly specialized alarm systems. There are key immune sensors involved, like RIG1, which spots specific RNA structures, and toll-like receptors, or TLRs, which sense different patterns in RNA. And when these receptors get tripped, they don't just sit there. They trigger the production of powerful molecules like type I interferons and other cytokines. These are basically antiviral agents designed to neutralize whatever threat it thinks it's found. And this is where the really groundbreaking work of Katalin Karikó and Drew Weissman comes in. Their breakthrough back in 2005 was just monumental. They noticed something really interesting. Unmodified mRNA, the synthetic kind, did trigger a strong immune response in certain immune cells. But eukaryotic mRNA, that's the mRNA naturally found in our cells, which has lots of subtle chemical modifications it didn't. That was the key insight. So they figured out critically that by incorporating specific modified nucleosides, things like pseudoridine or N1-methylpseudorin into the synthetic mRNA, they could essentially stop those inflammatory responses. Specifically, tweaking the uridine base seemed to be crucial, and this was like a double win, a huge deal. Because these modifications not only dramatically reduced the innate immune sensing, making the mRNA stealthy, but they also significantly enhanced its stability and how well it was translated into protein. N1-methylpseudoridine, for example, actually makes ribosomes stick around longer on the mRNA, so you get more protein. It was just transformative work and, absolutely deserving of the Nobel Prize they received for it.
That's genuinely fascinating because, like you said, for so long, triggering an immune response seemed like it should be a good thing for a vaccine, right? But you're saying that reducing that initial nonspecific immune flare-up was actually the key to making mRNA therapeutics work safely and effectively. It let the body focus its resources on this specific target, not just sound a general alarm.
Exactly that. That initial strong inflammation from unmodified mRNA would actually shut down protein production. It was counterproductive. By essentially, cloaking the mRNA from parts of the innate immune system, Karikó and Weissman unlocked its real therapeutic power.
Okay, so even with this incredibly clever modified mRNA, you still can't just inject it straight in, right? It's still fragile, it needs to get inside our cells. You need, like, a shield and a guide.
Yeah.
So how do these complex mRNA molecules actually get delivered safely? How do they get into the cells to deliver the instructions, past all the body's defenses?
Right, and this is where lipid nanoparticles, or LNPs, become absolutely essential. You really can't overstate their importance for the success of the COVID-19 mRNA vaccines. LNPs are basically tiny, tiny spherical packages. Think of them as microscopic delivery vehicles with a complex internal structure. They do two main jobs: protect the mRNA and transport it. They offer fantastic stability, they're biocompatible, meaning they generally get along well with the body, and they can carry a pretty hefty mRNA payload. Now, each LNP is typically made of four main types of lipids, all working together. First, and maybe most important, are the ionizable lipids. These are the real drivers. What's ingenious is their charge. They're positively charged at a low pH, like you find inside a cellular compartment called an endosome, which lets them grab onto the negatively charged mRNA. But crucially, they're neutral at the body's normal pH, like in your bloodstream. This makes them much safer and less likely to cause issues before they reach the target cell. This pH switch is key for trapping the mRNA and then releasing it later. Next, you have helper or neutral lipids. These guys help improve the particle's overall stability and the integrity of its membrane structure. One common one, DOPE, actually helps the LNP fuse with the endosome membrane, which is critical for the mRNA to escape into the cell's main compartment, the cytosol, where it can be read. The third ingredient is cholesterol. We all know cholesterol, right? Here, it provides vital structural stability. It makes the LNP membrane a bit stiffer, more robust. It also helps boost delivery effectiveness and makes the LNP last longer circulating in the body. And finally, there are PEG lipids. These are lipids with polyethylene glycol molecules attached. They sit on the LNP surface and create a kind of stealth coating. This PEG layer is really important. It helps reduce recognition by the host immune system, stops the LNPs from clumping together, and gives them more time to circulate and find their target cells.
That's incredible. So this complex four-part fatty bubble is basically what enabled those really successful vaccines like Pfizer-BioNTech's BNT162b2 and Moderna's Spikevax during the pandemic. I remember seeing the BNT162b2 trial results. 95% effective. That was huge. And the side effects were mostly mild short-term things like a sore arm, feeling tired.
Yeah.
So the LNPs really did their job getting those instructions delivered effectively.
They absolutely did. And what's interesting is that their role might be even more active than just being a delivery truck. LNPs themselves can also act as potential vaccine adjuvants. What that means is they don't just carry the mRNA, they actively help shape the specific immune response to the antigen encoded by the mRNA. They can stimulate certain innate immune pathways, which helps contribute to the strong immune activation that makes the vaccine so effective. So they're kind of part of the therapeutic effect, too.
Okay, so we've got the modified mRNA, which is super clever, and these brilliant LNP delivery systems. But science never really stands still, does it? It's always moving. How are researchers continuing to refine both the mRNA itself and the LNPs to make them even better, more effective, maybe safer?
That's a great point. And yeah, the field is constantly innovating. There's a huge amount of work going into mRNA sequence and structure optimization. For instance, scientists are meticulously tweaking the 5' and 3' UTRs, those untranslated bits we talked about, to really boost translation efficiency and make the mRNA last longer inside the cell. This involves things like avoiding certain RNA structures that can form, especially near the beginning, and making sure there aren't little accidental start signals called UTRs that could mess up the reading of the main message. Then within the coding sequence itself, there's codon optimization. Remember how the genetic code has some redundancy? Well, researchers swap out codons that are rarely used by our cells for ones that are much more common. This is based on which tRNA molecules are most available. It basically speeds up protein production and makes it more efficient. Often this means increasing the amount of guanine G and cytosine C bases while carefully managing the uridine content for stability. At the same time, there are huge improvements being made in how the mRNA is actually made, the in vitro transcription IVT process, and how it's purified afterwards. See, the IVT process can accidentally create unwanted RNA byproducts, especially double-stranded RNA, dsRNA, and RNA that hasn't been properly capped. These are major red flags for the immune system. So researchers are fine-tuning the IBT conditions, adjusting temperature, changing concentrations of things like magnesium, using those modified nucleotides during synthesis, all to minimize these contaminants. And after synthesis, really advanced purification methods are crucial. Techniques like HPLC, which is a type of chromatography, are used to get rid of that problematic dsRNA and other impurities. Plus, there's better capping technology. Using advanced cap analogs like the CleanCap system ensures that almost all the mRNA gets the right kind of cap structure, like Cap1 or Cap2, right during transcription. These caps look self to the cell, helping the mRNA avoid those initial immune alarms. And it's not just the mRNA. LNP design is also advancing rapidly. Scientists are developing new types of ionizable lipids. For example, branch-tail lipids might offer better stability and better fusogenicity. That's their ability to merge with cell membranes effectively to release the mRNA payload. And manufacturing is getting slicker too. They're moving from older methods to techniques like microfluidics mixing. This allows for the production of very uniform, precisely sized, and highly stable LNPs much more reliably and scalably. And finally, for some applications like certain vaccines, you actually want a specific controlled immune boost. So scientists are exploring ways to add defined innate immune adjuvant activities. This could involve attaching small immune-stimulating molecules directly to the mRNA, ensuring they get delivered together to the right cells to fine-tune the immune response. It's really getting quite sophisticated.
Wow. So what does all this relentless optimization mean for the future? I mean, this engineering feat was obviously critical during the pandemic, but where else is this incredibly versatile mRNA platform taking medicine? It really feels like the COVID vaccines were just scratching the surface.
Oh, they absolutely were. The potential is enormous, and it's expanding incredibly fast. Beyond just preventing infectious diseases, one of the most fascinating areas is inducing immune tolerance. This is almost the flip side of vaccination. Instead of boosting immunity, the idea is to use modified mRNA LNPs to suppress overactive immune responses. Think about treating allergies. There are approaches being developed, for instance, for peanut anaphylaxis, where mRNA delivers specific components to liver cells. This encourages the development of regulatory T cells, or TREGs, which then calm down the allergic reaction. Similarly, it's being explored for autoimmune diseases, like in models of multiple sclerosis. Here, highly purified, modified mRNA is used to deliver antigens in a way that promotes tolerance, dialing down inflammation by inducing those TREGs rather than activating aggressive immune cells. We're also seeing huge strides in cancer immunotherapy. This includes developing personalized cancer vaccines. These can be tailored to target unique mutations or neoantigens found only in an individual's tumor. Plus, mRNA can be used to make immune cells express specific proteins that help them better recognize and destroy cancer cells. Protein replacement therapy is another really exciting avenue. If someone has a genetic disorder where they're missing a crucial protein, or don't make enough of it, mRNA could deliver the instructions for cells to produce that protein. Think factor VIII for hemophilia, for example. It offers a whole new way to treat these kinds of conditions. And maybe one of the most revolutionary aspects, especially for global health, is the push towards decentralized and mobile manufacturing. Imagine being able to produce mRNA vaccines right where they're needed, quickly, instead of relying on complex centralized factories and cold chains. You've got initiatives like BioNTech's BioNtainer. These are literally self-contained manufacturing labs built into standard shipping containers. They're designed for rapid, scalable production, potentially making millions of doses a year. They're being deployed in places like Africa to tackle vaccine inequity head-on. There are other similar concepts too, like mobile on-demand units, all focused on bringing production closer to the point of need, even in remote areas. And looking even further ahead, there are emerging RNA technologies that could push the boundaries even more. People are exploring circular RNA or circRNA. Because it's circular, it might last longer in cells and produce protein for longer than standard linear mRNA. And then there's self-amplifying RNA, or SARNA. This type of RNA includes extra instructions that tell the cell to make many copies of the therapeutic RNA message itself. This means you could potentially use a much lower dose to get a really robust response, which could be great for vaccines.
It's just an incredible range of possibilities, isn't it? It truly feels like a medical revolution in progress. Of course, with such fast innovation, there are always going to be lingering challenges. We still need to figure out things like maintaining cold chains everywhere, fully understanding rare side effects, things like myocarditis or pericarditis that we're seeing, albeit rarely, with the COVID vaccines. And really nailing down the biodistribution, where exactly does the mRNA LNP go in the body and how long does it stick around? But yeah, the overall direction is clearly forward and it's moving fast.
Absolutely. And, it raises a really interesting question for the future. We've seen how meticulously tailoring every single aspect of mRNA, from the bases themselves to the delivery package, has led to these incredibly powerful new medicines. But as we gain even more precise molecular control, how might our growing understanding of that subtle balance, the balance between activating the immune system when we need to and suppressing it when it's overactive, allow us to treat an even wider spectrum of diseases, perhaps even conditions that today we can barely even imagine treating effectively?
There's a fascinating thought to end on. The mRNA revolution really does feel like it's just beginning, and the deeper we dive into this incredible science, the more potential we seem to uncover. Makes you wonder, what will you discover next about this incredible science?